Hi everyone, welcome back. I decided because these lectures are mad long that the pre-recorded version is probably a better bet to go for lecture 7 and lecture 8. We did lecture 6 live, but yeah, it's just a lot of material to get through, and I feel like for you guys to be able to digest the material, be able to pause and take breaks, the pre-recorded way may be the way to go. So we're going to do that. Let's get into it. We're going to do the second to the last lecture today, which is lecture 7. So let me share my screen per usual. Okay. And all right, great. So you guys should be able to see that. And let me make this a little bit bigger. Whoa, that's a little big. That's okay. Okay. So yeah, lecture 7, I can't believe we've gotten this far, but we have one more lecture after this. Yay. But also sad. As you guys know, you can reach me at this email address anytime you need me. All right, so last class we took a journey and we actually covered one of the more difficult concepts for this course, which is alternation of generations. And in order for us to understand alternation of generations, we had to talk about cell division, mitosis, and meiosis, right? If that still seems a little cloudy to you, that's okay. I think today might help reaffirm some of these concepts. We'll see a lot of repetition in terms that we used last class. But briefly, mitosis is a type of cell division, and meiosis is a type of cell division. Those are two types of cell division. Mitosis is for body cells, and mitosis is used to make new cells, new body cells, whether it's to grow part of the body bigger or to repair part of the body that has been damaged, right? That's what mitosis is for. And when you go through mitosis, you have one cell that divides into two cells, and those two cells are still diploid. The original cell was diploid, and those two cells are identical to each other and the original cell. Meiosis, on the other hand, as we talked about, but this is a little review, is for the sex cells. In humans, meiosis leads directly to gametes, egg and sperm. But in plants, because plants are different, they have two different organisms, and meiosis actually leads to the production of spores, and those spores are haploid, and those spores will grow into gametophyte plants. The gametophyte plants will in turn make gametes, egg and sperm, and then when egg and sperm unite, you're back to your diploid sporophyte plant. So when meiosis occurs, there's two stages of meiosis, which you don't need to know, but the point is when meiosis occurs, you start with a diploid cell and you end up with 4 haploid or 1N cells. All right. So we talked about alternation of generations in general, and we also talked about the process in these non-vascular plants, right? The mosses, liverworts, and hornworts. And then we talked briefly about asexual reproduction in those plants. Today, we're going to continue this discussion of alternation of generation, but we're going to be talking about it in vascular plants. In particular, we're going to start with the spore-shedding vascular plants, we're only going to cover lycopods and ferns, even though your book covers a few others. And then at the end, we'll talk about seed-shedding vascular plants, gymnosperms, which include the cycads, ginkgo, and conifers. So vascular plants. Well, last time we talked about vascular, we talked about non-vascular plants. And when we talk about non-vascular plants, there's a reason why we call them non-vascular, right? And it's because they lack or don't have vascular system. They don't have xylem and phloem. But vascular plants, surprise, have xylem and phloem, and that's why we call them vascular plants. So just to compare non-vascular plants and vascular plants. So last time we talked about non-vascular plants, as I just said, non-vascular plants don't have any vascular tissue, no xylem and phloem. Non-vascular plants also have free living gametophytes that are dominant. So when you see a moss, or you see a liverwort, or you see a hornwort, you're usually looking at the gametophyte plant because that's the dominant generation. And since the gametophyte plant is a dominant generation, the sporophyte plant is very, very reduced and actually has to be attached to the gametophyte plant for its whole part of its journey. So sporophyte plant in non-vascular plants has to be attached to the gametophyte plant. It gets all of its nutrients usually from the gametophyte plant. Vascular plants are different. Vascular plants have, for one, vascular tissue. That should be an easy thing to remember. Non-vascular plants, no vascular tissue. Vascular plants have vascular tissue. That should be easy enough. And then the other things are actually the opposite of what we saw in the non-vascular plants. So in the non-vascular plants, we had free living gametophytes that were dominant. In the vascular plants, we have free living sporophytes that are dominant. So when you look at a fern, or you look at a lycopod, or you look at a conifer, what you're seeing with your eyes is the sporophyte generation. And on those sporophyte plants, the sporangia is now going to be attached to leaves, Remember, in the non-vascular plants, the sporophyte plant often emerged from the gametophyte plant sometimes having a foot, a seta, and a capsule in the case of the mosses, sometimes having a foot, a seta, and a capsule in the case of the liverworts. And then in the case of the hornworts, you had a foot, you had that basal meristem, and then the capsule, right? Now, sporangia, or capsule, is going to be attached to leaves of the sporophyte plants. And the gametophyte plant is going to be very reduced. So only lasting a very short period of time and being very, very, very small. Often visible with the naked eye, but often needing a microscope to see it. Okay, depends on who we're talking about, right? In the ferns, you can see the gametophyte with the naked eye. When you get to your cycads and your conifers later in this class, you would need at least a dissecting microscope to see the structures. Okay, so here's our roadmap that we've seen before. We've already gone through those non-vascular plants that are at the bottom. Now we're in the vascular plant part of the plant kingdom, right? Here we are, we're here, and we're going to start our journey here in these spore-bearing, spore-shedding vascular plants. Oh, look at that. Here we are, spore-shedding vascular plants. Ferns, lycopods are who we're going to talk about today. The book covers a few more, but we're only going to, for the purposes of this class, ferns and lycopods are plenty. So lycopods. Lycopods, if you live in New Jersey, you've probably seen them before and not even known that you've seen them before. They're very common in kind of wooded areas. And maybe to the untrained eye, they look kind of mossy, right? They look kind of mossy. In fact, one of the common names of them is club moss. So yeah, they look a little mossy, but they're not mosses because they have vascular system, among other things we just talked about. Two types we're going to learn about in this class are Lycopodium, which is shown at the top here. Oh, there we go. So the one here shows Lycopodium, and the one up there shows Lycopodium. And the other one we're going to learn about is this one called Celogenella. So Lycopodium and Celogenella are both lycopods. Lycopodium and Celogenella are both lycopods, right? Lycopods. Can't draw to save my life. All right. So those are the two we're going to learn in the class. And again, you guys don't need to know the phylum names, right? You don't need to know Lycophyta, and you don't need to know Polypodiophyta. But you'll see those terms in the book, which is why I'm including them here. But for the purposes of this class, knowing lycopods is fine. But you will need to know Lycopodium versus Celogenella, because they're both lycopods, and that's the only other way we can distinguish between the two of them. And then ferns. So ferns are also spore-shedding plants that are vascular. And here's a little group of ferns. I actually, for the purposes of this class, I broke out some of my ferns. Ooh, so that you can see real fern and real life fern. Oh, you got another one here. Ooh, pretty nice. Yeah, so we'll talk about ferns. Other things mentioned in the book which we won't cover are wisp ferns, horsetails, and quillworts. You should at least know that there are other spore-shedding plants other than just lycopods and ferns, but we won't cover these others in detail. Okay, so the spore-shedding vascular plants. First, we'll start with lycopods. So here is, on the top, you have different Lycopodium, and on the bottom here, you have different Celogenella, both of which are lycopods. And the reason lycopods are called lycopods is because whoever discovered them originally thought that they looked like a wolf's foot. So leukos comes from the Greek. Lyco comes from leukos, which is Greek for wolf. And then pod comes from pous, which means foot. So whoever first discovered these thought, or described them, was like, this looks like a wolf's foot. It is the oldest surviving group of plants, which is super, super cool. Okay, oldest surviving group of vascular plants. What that means is these plants have been around in some shape or form since around 400 million years ago in the Devonian era. And they are, to our knowledge, the oldest remaining lineage of vascular plants on Earth today. Pretty cool. There's about 800 species that are still around today, so that's what extant means. Maybe you've heard the word extinct before, like this animal is extinct. You can see it here, extinct. Extinct means it used to be here and it's no longer here on Earth, whereas extant means still here on Earth. So there's about 800 species that are still here on Earth, and because it's a vascular plant, we know that the Sporophyte generation is dominant. And so, yeah, like somebody's walking around and they're trying to describe all these plants, and they see this plant and they say, oh, it's a wolf's foot. And like maybe a little bit, like here's wolf's feet on the right. I mean, yeah, like a little bit, but like if we get a little creative, okay, sure. Like I can see that looking. There must have been a lot of wolves wherever this person described these, and I forget where that was now, but since it's from Greek, I'm guessing around there, but who knows. So with your, remember, where knowing Lycopod is fine, right? Lycopod for describing these both. But there's two genera that we will learn about: Lycopodium, of which there's about 50 species, and Celogenella, of which there's around 700 species. Both of them occur in tropical environments usually, like I said, there's plenty of lycopods that occur in even like the New Jersey, Connecticut, New York region in the woods. So they can live in temperate areas as well. And the sporophyte plants, which are dominant because we're in vascular plants, can either be terrestrial, which means grow in the ground, or epiphytic, which means they are epiphytes, which means that they can grow on other plants, which we also came across in the non-vascular plant lecture. I'll just write it here, epiphyte. If you're an epiphyte, it means you're a plant that can grow on other plants, usually trees. So, oops, no. Can I grab that? Oh boy, this is why. Okay. Undo. Redo. Epiphytes, plant that can grow on other plants. And I think I can, select. I can select it. So when I say down here that sporophytes can be terrestrial, which means grows in the dirt on land, or epiphytic, it just means that they grow as epiphytes, which are plants that can grow in other plants. Okay. So here you go. On the left is a picture I took years ago in a forest in, I think, Massachusetts when I was doing field work. and I saw these little things coming out of the ground. It almost looked like somebody had taken like a Christmas tree and buried it underground, and it was still green. Like that's literally how it looked. And at the time I had no idea what it was. This was years and years ago. So I took this photo because I was just very confused. But I found out later that those are types of lycopods. And then, so those are growing as terrestrial lycopods, and the ones on the right here are growing as epiphytic lycopods. Here's Celogenella. Oh, those were Lycopodium. And here's examples with Celogenella. On the left, we have Celogenella growing in the ground, terrestrially. And then Celogenella growing on a tree as an epiphyte, right? Or has an epiphytic habit. Epiphytic habit. Okay. Parts of the sporophyte plant. What's dominant, because we're in the vascular plants now. So now we're vascular. We have roots, stems, and leaves. The non-vascular plants don't have those because they don't have vascular tissue. So you can't have roots, can't have stems, can't have leaves unless you have xylem and phloem. In the lycopods, the roots are what we call adventitious, which means that they grow off the stem in random places. The stems are what are called rhizomes, which is a fancy word for horizontal stem. Here, I'll try this again. Let's see if I can do this. Let's move this away. Okay, adventitious means roots grow off the stem. All over the stem. Not just like at the base, they grow all over the stem. Oops. Rhizomes are horizontal stems, which makes sense if the roots are adventitious, because basically the stem grows horizontally and roots just come out of it wherever it's touching dirt. And then lastly, microfils. Microfils is a new term for us. Microfils are simple leaves. Remember simple versus compound leaves in the leaf lecture. Go back to that if you need a refresh. But microfils are simple leaves that have one vein. Simple leaves that have one vein. Okay. So here you go. These are microfils. Maybe my head is blocking the thing, but what it says is leaves are simple in that they have one vein, aka microfill. And so so here you go. On the left, you have a stem, and all those little pokies coming off the stem are microfills. This stem is from Lycopodium, and all those little pokies are microfills. I will say, so microfill is one vein leaf, right? Fill means a leaf. Fill always means a leaf, by the way, when you see like other things ending in fill. And micro in this case means a one-veined leaf. But I want to point out that later in this class and then going forward, micro is going to refer to a different thing. So just try to keep that in mind and pay real attention to detail when we switch to that next thing. So for microfill means a simple leaf with one vein. That's what microfill means. Like write it down and underline it somewhere. My head also might be blocking this. But so you have microfills, which are a simple leaf with one vein. And some microfills have sporangia on them, because I said in the top of the class that in the vascular plants, sporangia are located on leaves. And in the case of the lycopods, Lycopodium and Celogenella, the sporangia are located on microfills, right? Simple leaves with one vein. If a microfill has a sporangia on it, my head might be blocking it, but they're called sporophylls. So if a microfill has a sporangia on it, we call it a sporophyll. In that case, fill still refers to leaf, and sporo, as you should be familiar now with the term sporophyte, right? Sporo refers to spores. So a sporophyll means a leaf that bears or makes spores. Okay. Sporophylls. Special leaves that bear sporangia. Sporo equals spores, and phyll is leaf. In the Lycopods, Intelligenella and Lycopodium, sometimes the sporophylls look like microfills. Like sometimes they look the same as the microfills that don't have any sporangia on them. And in that case, they'll be green and photosynthetic and look the same. Other times, the sporophylls are a little different looking in that they're smaller, not green, and arranged into a structure, a new structure for us, which is strobilus, and the plural is strobili. This is another term that, even though right now might seem scary, we're going to encounter a lot of times today. So you'll have a lot of repetition. So this is what I was talking about on the previous slide. Maybe my head is blocking it. It says, Figure 1, sporophylls are identical to photosynthetic leaves. So if you look on the picture on the left, the green leaves look just like, here I'll use black. All of these green leaves look just like regular microfills. And these little yellow things at the base of them are the sporangia. But they just look like normal microfills. They're green, nothing fancy. They're not organized in any special structure. They're just regular green microfills that happen to have sporangia on them. Whereas in figure 2, this is an example of the sporophylls that are morphologically different than microfills that don't have sporangia on them. So, and we can see both in this figure. So at the top here, you might notice these kind of clusters, right? And those things are called strobilus. One of those is a strobilus. So here, I'll do it again. Here is one strobilus. Here are 3 strobili, which is the plural. And in that case, if you look at it, the leaves don't look like normal green leaves. They're not green. They're kind of yellowish in this picture. They're smaller because they have to fit onto a strobilus. And then they're organized into a strobilus. To compare what I'm talking about, if you look down here, this is on the same plant. These leaves are green. These leaves are not organized into a strobilus. These leaves down here are just regular microfills. They don't have any sporangia on them, whereas all the sporangia are located of leaves that are in a strobilus. Okay. This is the same thing again, but a different way to look at it. So on the left here, that is like your figure 1, right? That's like your figure 1 on the left here. And you notice like some of the leaves toward the top of that plant, they don't have sporangia on them. So we just call them regular microfill because they don't have any sporangia on them. But below those are some microfills that happen to have sporangia on them. And so we call them a sporophyll. Remember, phyll is leaf. So microphyll, leaf with 1 vein. Sporophyll, a microphyll with a sporangia on it. In this here, those little yellow things are the sporangia. Over here, you can see the sporophyll leaf is right here. And there is a sporangia on it, sporangium on it. And inside that sporangium is going to be where spores are made. That inside that sporangium is also where meiosis is going to occur. Because remember, this whole thing here is Sporophyte plant 2N. Sporophyte plant makes what? It's a plant or a phyte that makes spores, sporophyte. The way it makes them is via meiosis, and those spores that it's going to make, which are in the sporangium, are going to be haploid, right? That's what we talked about last time during alternation of generations. Same thing, just a better microscope slide. So here on the left is the same thing that we saw. You see here it's saying sporophyll. I'll use a different color. I'll use dark blue. So sporophyll. So here is one sporophyll. Here's another sporophyll. They're attached to the stem, sporophyll. And on top of those sporophylls, there are sporangia, which are these blobs. And inside of the sporangia are going to be where meiosis occurs to make haploid spores. And the haploid spores are all those purple dots on the left. Here's a close-up of one on the right. Here's the sporophyll, right? The leaf the sporangium is attached to. Here is the sporangium. And all of those purple things inside are spores. My head is blocking the word spores, most likely. Okay. Now we know a bit of the terrain. Now we can speak more specifically a little bit about Lycopodium and Celogenella, both of which are lycopods. So we'll start with Lycopodium. Here's what they look like. Remember I said before, to the untrained eye, they kind of look like a moss? Yeah, well, they do, and the common names often are something with the word moss in it. They are not mosses, they're vascular plants, but sometimes, you know, the common name sticks. So here's two examples of Lycopodium that actually occur in New Jersey, New York, Connecticut region. Yeah, those are examples. Sporophyte, right, which is the dominant generation in Lycopodium and all lycopods and all vascular plants. So that's what the sporophyte plants look like. Let's look a little bit at what the gametophyte plants look like. So this is something we're going to come across today. We have to kind of keep track of because we didn't talk about it last time at all. In that some plants make one type of spore, and some plants make two types of spores. All the non-vascular plants, the mosses, liverworts, and hornworts, only make one type of spore, which is why we don't even mention it back then, because they all make one type. Some of the plants we're going to come across today make two types. But Lycopodium, just like the non-vascular plants, only makes one type of spore, one spore type. And that spore is going to grow. When you hear the word grow, bing, bing, things should be going off in your brain when you hear the word grow. When you hear the word grow, grow, you say? Oops, grow? What grow? Mitosis, 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 mitosis. Right? So if you hear the word grow, you should automatically be like, oh, right, mitosis. But Lycopodium makes one type of spore. The sporophytes make one type of spore. That spore is going to grow via mitosis, right, into gametophyte plants. And the gametophyte plants are going to grow outside of the spore wall. That's what it says behind my head, or it says outside of the spore wall, same thing. And that's just like we saw in the non-vascular plants. Remember? In the non-vascular plants, when you see the plant, you're looking at the gametophyte generation. So of course it's growing outside of the spore wall, right? Because we see a big old fluffy moss. Well, in the Lycopodium, the gametophytes don't get quite as big as we saw in the non-vascular plants, but they still grow outside of the spore wall. So like the spore will land on dirt and then the gametophyte plant will grow out of it, out of it. Here are two examples of Lycopodium gametophyte plants. On the left, you have kind of like green blob, right, which is photosynthetic. And that one will just land on the surface and grow. On the one on the right, it's from a different species. It's not green, kind of almost looks like a little radish or something. That one will have an association with fungus in the soil in order to get nutrients. But either way, those are gametophytes of Lycopodium. Very small, very, very small, like smaller than a dime. See, so they're small. And note, the gametophyte plants are still non-vascular because they're that small. And they don't live that long. So the gametophyte plants are small, the gametophyte plants are non-vascular, and the gametophyte plant have these things called rhizoids, which are hard to see, maybe on this and which we kind of didn't talk too much about last class, but rhizoids are kind of like little hairs, which we don't really see well in this picture, that come off of the gametophyte plants, and they just serve to kind of anchor it to the soil. If you're in the lab, you may have come across the term rhizoids as well. And this is what I was just saying on the previous, a couple seconds ago, that depending on the species, you have different morphologies. Sometimes you're photosynthetic, like the one on the left, and sometimes you're not photosynthetic, like the one on the right. So these are the Lycopodium gametophytes. Now we're going to go through the Lycopodium alternation of generations. And this is good because there's going to be a lot of these repeat terms, and it'll help you guys remember it. That's the only way to remember these things, just say them over and over and over again. So let's do it. So let's see, what color should I use? I'll use pink. So here you go. This right here, this whole thing is a sporophyte plant. Lycopodium sporophyte plant. 2N. See? Mature sporophyte. 2N. Which is the dominant generation in Lycopodium and all vascular plants. So here's some of the things that I just already have mentioned, right? You have rhizome, I'll color it here, rhizome, which is a horizontal stem. And then you have roots coming off of this horizontal stem. And because they're coming off of the horizontal stem, we call them adventitious roots, as we said on the previous slide. This sporophyte plant is the result of egg and sperm coming together, right? And what made the egg and sperm? Well, the plant that makes gametes, the gametophyte plant makes egg and sperm. So egg and sperm would have come together on that gametophyte plant. Now we see the mature sporophyte plant is emerging out of that gametophyte plant, right? Which, depending on the species, may be a green blob or look kind of like a radish. In the case of this one here, it's more like the radishy kind of gametophyte plant we saw in the previous slides. We're talking about Lycopodium, which is a lycopod. So the leaves are microfils, right? A leaf that's simple with one vein. However, at the top, in this case, at the top of the stem, are these structures called strobili, which we just learned are where there are leaves that have sporangia on them. And if you're a leaf that has sporangia on it, in the case of the Lycopodium, it's called a Sporophyll, right? Which is a microfil with a sporangium on it. So this is showing you that down here where there's no sporangia on the leaves, we just call them regular old microfills. But once they have sporangium on them, call them a sporophyll. So here we're doing a close-up of the tip of this strobilus. And you got a bunch of sporophylls here. All of these things here are sporophylls. All of them are a microfill that has a sporangia on it. Now we do a close-up of one of those one sporangium. And if we look inside, we see that there is 2N tissue, which we've been calling spore mother cells, but diploid tissue that's going to undergo meiosis to make spores. Okay, this is what we just saw. To show you what we're talking about in real. Here's a reminder, right? Okay, so this is where we just left off, right? We were looking at our close-up of our sporangium, and inside are these 2N spore mother cells that are going to undergo what? Meiosis. Every time you undergo meiosis, you go from a diploid or 2N thing to a haploid or 1N thing. And because we are talking about plants, I don't know if you guys can see it that well, but you'll be able to access these slides. Anyways, once meiosis occurs inside of that sporangium, the result is, so here is that sporangium again, and meiosis has already occurred, and the result is these 1N spores. 1N spores. Those haploid spores are going to leave the sporangia, and look, here's a sporophyll right there. But they're going to leave the sporangium to grow into gametophyte plants. So you see there, germinating spore. Notice on the germinating spore, look, see this here, I'll use green. See this like thing coming out? That's the gametophyte plant starting to grow, mitosis, grow out of the spore. Okay, so here you go. Gametophyte plant is growing outside of the spore. See, young gametophyte, that's this. Growing outside of the spore, we see these little things I just mentioned, these little hair-like things called rhizoids that help the gametophyte plant stay anchored to the soil. Don't get confused with rhizoids and rhizomes, just because they start with the same 3 letters. Rhizoid is little hairs that emerge from the gametophyte and anchor it to soil. Rhizome, horizontal stem of a sporophyte plant. Don't get these 2 terms confused just because they start with the same 3 letters, okay? Sometimes that trips students up. All right, so yeah, so little gametophyte, whoa. Oh my God. Hold on, I got a tornado warning. I'm gonna pause. Okay. Oh, that's a first. Everything's fine. Yeah, so basically what happened was my phone started making crazy noises, and it was because there is a it's because there was a tornado not too far from my apartment in Michigan. And so it had advised that I go into a room with no windows and things like that. Super fun thing to experience. But yeah, so that's what that was. I hope that never happens again. I would bet money that that would never happen again while lecturing, but okay. Let's shake that off a little bit. Never a dull moment. So I'm going to regroup and let's get back to where we were, which was me basically yelling at you guys about not confusing rhizome and rhizoid. Let me make this big. Yeah. Yeah, apparently it's been raining here for days, and like apparently there's a ton of flooding where people are just abandoning cars, hundreds of cars on the highways in and around Detroit. That's just been weather-wise been really, really crazy here, which I guess is a thing that happens, but always kind of throws you for a loop. Let's see if I can get this lighting better, but maybe not. Okay. All right, so back up a little bit if you want to get oriented, but basically where we left off is we have this 1N haploid spore, which was the result of meiosis inside of the sporangia. Now the spore is germinating in that the gametophyte plant is going to grow outside of the spore. So here is that young gametophyte plant. It's emerging from the spore, right? It's coming out of the spore. And I was saying these little hair-like things called rhizoids are going to help the gametophyte plant attach to the surface of the soil or a rock or whatever it's going to be attaching to. So now we have a close-up of a gametophyte plant. This is a gametophyte plant of Lycopodium. And usually when plants grow outside of the spore, as we have here, usually you'll have both sex organs on one plant. So usually they will be you could say bisexual, or you could say that they are monoecious and that they have both sex organs in one plant, right? And that's the case here. So we should already know now, based on kind of going through this a few times, that the archegonia is going to have inside of it an egg, and the antheridia is going to have inside of it tissue that's going to make sperm, right? Tissue that's going to make sperm. Let's get out of this. Right, and so this is just to show you here, if my head is blocking it, you'll be able to see it on the full slides that are posted on Canvas. But basically it says, note, the sporophyte plant produces one spore type, right? Which is something called Homosporous. Your book also has it as well. I'll write it right here. Homosporous. Homosporous means sporophyte makes one type of spore. Okay, so that's what Homosporous means. One type of spore. And when you have Homosporous spores, the gametophyte always is going to grow outside of the spore wall. So every time you have Homosporous or one type of spore made by the sporophyte plant, gametophyte grows outside of the spore. And that's what it says behind my head in case that my head is blocking it. And as I just said, the gametophyte that grows outside of the spore pretty much is always bisexual or monoecious in that it has both male and female sex organs on it. So these little prompts will help you when you guys are doing this by yourself. So this on the right is our male sex organ, the antheridium. And inside of it is tissue that is tissue that's haploid that's going to make sperm, because everything on the gametophyte is haploid. And so eventually the antheridium will mature and be able to release sperm. You can see the little sperm swimming out. Here is one that they've circled, and here it is big. This is one Lycopodium sperm. Sperm is going to swim to the egg, just like we've seen before, right? Here's egg. Egg is inside of what sex organ? Archegonium. So here you go, sperm swim, swim, swim to unite with the egg. Once the sperm unites with the egg, notice the color change on this slide, right? It goes from orangey color egg to a green color zygote, right? Because we had a haploid sperm, haploid egg fused together during process of fertilization, and we end up with a diploid or 2N zygote. But hopefully these things are starting to seem more familiar to you since we've gone through them a couple times, and we will go through them even more. But here's our 2N zygote on the right. That is the beginning of your sporophyte generation. Trying to think before I use kind of like goldish color for gametophyte and bluish purpley color for sporophyte. But here you go. Here's your 2N zygote, still located in that archegonium of the gametophyte plant because that's where the egg was. Then the zygote is going to grow, mitosis, into a young embryo. And then that young embryo is going to grow into eventually the mature sporophyte plant that has all of those things that I've already mentioned, right? A horizontal stem or a rhizome, roots that come off the stem, adventitious roots, and then microfill leaves and strobili that have microfill leaves that have sporangia on them, which we call sporophylls. Okay. That's Lycopodium, which is Lycopod, which is a vascular plant, which is a spore shedding vascular plant. So now we're still talking about the Lycopods, but this is another member, which is called Celogenella. So Celogenella is super cool. And I wanted to highlight this one species of Celogenella. And so I'll stop sharing. So look, you can see here, Celogenella lepidophylla, or the resurrection plant. And so check this out. This is a type of Celogenella that lives in the desert. And as you may know, you do not get a lot of rain in the desert. And so what happens is this plant, the Sporophyte plant, actually kind of hangs out in this dried up ball form. So that's the Sporophyte plant. It's kind of like all dried up. It almost looks dead. Almost looks dead to the naked eye. But when the rains finally do come in the desert, what looks like a dead Celogenella plant will actually resurrect and within a few hours look like this. And so of course, because this is just too cool, I have a video of that right here, which I'll share, and I'll make it big. So this is one of those Celogenella plants that's been all dried up. And so somebody basically just took it and put it in a bowl of water. So watch what happens. Look at this. You can see there's a timer in the bottom there in minutes, 18 minutes, 19 minutes of being in the water. And check it out. Look at that. What? So this little dried up ball of Celogenella gets exposed to water and immediately erupts into this beautiful green plant. And so what it's going to do though is it's going to have to complete its life, like it's going to have to do stuff real quick right away, because it's not going to have that going on too long in terms of, you know, the rain's not going to be there that long. So if it needs to make spores really quickly, and then those spores need to get in the soil really quickly, and then those spores need to germinate into a gametophyte plant really quickly because the sperm's going to need water to swim. So it has to happen super, super fast in order to make the next Sporophyte generation before the rain stops. Super cool. Whoops. Oh, there I am. Yeah, so, and just so you know, this is a type of Lycopodium, and it also has Strobilia, right? So, and it also has Microfils. So the leaves down here are just regular Microfils. And then the leaves that are part of the Strobilia are Microfils that have sporangia on them, hence Sporophylls, right? Okay. And you can see in this little map, these occur in south parts of the US. Okay. Finally we get to this concept. So all the things we've looked at so far, mosses, liverworts, hornworts, and Lycopodium, produce one type of spore. Sporophytes produce one type of spore. Now, in Celogenella, the Sporophyte plants produce two types of spores. And in terms of plant evolution, this is a big deal. This is a super big deal, and I'll explain why. So now we have two types of spores, and we can talk about them as being microspores and megaspores. And so you may have remembered back when I first introduced the concept of a microphyll, I said microphyll is a simple leaf that has one vein. But I said later in the class, we're going to encounter this prefix micro again, and it's going to refer to something different. And what it refers to now is male, So microspores are going to grow into male gametophytes, and megaspores are going to grow into female gametophytes. Every time going forward now, except for microphyll, every time you see the prefix micro or mega, micro always going to refer to male, mega is always going to refer to female. And the reason that is because in plants, the female structures tend to be much larger than the male structures, hence the mega versus micro. So now, though, another difference is we have two types of spores. So we have spores that are going to make male gametophyte plants and spores that are going to make female gametophyte plants. But in addition, another difference is that now the gametophytes are going to stay inside the spore. So anytime you have a sporophyte plant that makes two types of spores, the gametophytes are always going to stay inside of that spore. So the male gametophytes are going to be inside of the microspores. The female gametophytes are going to be inside of those megaspores. Here is a picture to show you what I'm talking about. So on the left here, you have a megaspore, which is this big giant ball. And all these little dusties around it are the microspores. And so notice the size difference, hence why they are called microspores, and hence why the female, the spore that grows the female gametophyte is called megaspore. So there's one male gametophyte plant per microspore, and there's one female gametophyte plant per megaspore. This is what I had just said. And just so you know, and we will see it on slides coming up, just so you know, move this out of the way. When you have sporophytes that produce two types of spore, they are called heterosporous. So those sporophytes are heterosporous. Hetero means different, and sporous refers to spores. So they're sporophytes that produce two different spores, the microspore and the megaspore. Remember that when the sporophyte plants produce one type of spore, we call it homosporous. Homo means same. So they produce all the same spores. One type. Okay. So when you have gametophyte plants that are growing inside of spores, each gametophyte plant has either male or female sex organs on it, and therefore the gametophyte plants are dioecious or unisexual. So here you go. This is a drawing of a megaspore with the female gametophyte plant growing inside the megaspore. And the female gametophyte plant, even though it's inside a megaspore, is still going to have on it archegonia. So just to get you oriented here, this yellow that I'm drawing here is the megaspore wall. So you can see all that stuff. And then inside is this female gametophyte plant. It grows inside. Now the female gametophyte plant does poke out a tiny bit, and that's so that sperm eventually can meet her and fertilize the eggs. But she stays inside the megaspore wall. And the male gametophyte plant grows inside, excuse me, inside of the microspore. And the male gametophyte plant, in order to fit inside of that microspore, because you saw how small they are, right? You saw how small these microspores are. In order to fit inside, the male gametophyte plant is basically just a single antheridium now. That's all that remains of the male gametophyte plant. So you see a very large reduction in both gametophyte plants. So I'm drawing the microspore wall. And inside of the microspore is a single antheridium that will make sperm. And that's it. That's it. So in both cases, we have a really dramatic difference in size compared to when we were talking about, the mosses and liverworts and hornworts, where everything you see of them is basically the gametophyte plant, right? Green fluffy, that's the gametophyte plant. Well, now gametophyte plant's so small, stays inside of a spore. So large reduction in size, particularly for the male gametophyte plant, which is now just a sex organ, basically, just an antheridium in each one. Okay, so we have to go through the alternation of generations in Intelligenella, and a lot of these terms will be repeat terms that we're already used to seeing, with a few exceptions. So here is our, I'll use blue for the diploid parts. Yes, so here, what's happening down here is the female gametophyte plant has already been fertilized. Her egg's already been fertilized by sperm. And so you have egg and sperm, which are both haploid, unite to make your diploid zygote, which is the beginning of your sporophyte plant. And so now we see that sporophyte plant emerging out of where the egg was, right, in the archegonium of the female gametophyte plant. Eventually the wall of the spore will just fall off gametophyte will disintegrate. And the sporophyte plant, which is this, remember, which is this thing, will be able to just go live its life independently because it's the dominant generation. So here we see this whole thing here is the mature sporophyte plant of Celogenella. You know, it's got stems, it has microfills, right? Leaves that are microfills. It has regular roots that are adventitious because they come off the stem, just like in the Lycopodium. Has strobilus or many strobili, right? That's the plural. And on the strobilus is the same thing as we saw in Lycopodium, microfills with sporangia on them, aka sporophylls, right? So now we're going to look at a close-up, close-up of, so you see strobilus on top. So here's a strobilus close-up. And this is Celogenella. So remember, we have two types of spores that are made by Celogenella, right? We have Microspore that has the male gametophyte inside and Megaspore that has female gametophyte inside. Since we have two different types of spores, we have two different types of sporangia that make each of them. And remember, micro refers to male and mega refers to female. So the microspore is going to be made in a sporangium that's called the, you might have guessed it, microsporangium. That's where the microspores are going to be made. That's where meiosis is going to occur to produce spores, in this case, microspores. The megaspores are going to be made in a sporangium that's called a megasporangium. And likewise, now that we have two different types of sporangia, we can get more specific about how we describe the leaves they're on. This is where it's helpful to keep these prefixes in mind. So a megasporangium that makes megaspores is on a leaf called a Megasporophil. And here's another microsporangium here. Microsporangium is going to make microspores, is going to be on a leaf called a Microsporophil. So these terms, sporangium, spore, sporophyll, all we're doing is adding the prefix micro or mega, depending on which one you're talking about. Now it's raining again really, really hard. If my phone goes off again, I may have to go back into the bathroom for a while and hide. All right. So hopefully it doesn't. So yeah, so just so when you're looking at these slides on your own, if you can see the rain. Not really. Just so when you're looking at these slides on your own, here you go, microsporophyll, megasporophyll, and the terms that I just described, right? So you don't get confused. All right. So microsporophyll, sorry, microsporangium is going to make microspores. Megasporangium is going to make megaspores. And the female and male gametophyte plants are in those spores, right? So here we go. Here is let's do megaspore first. Here's a megaspore. Inside the megaspore is the female gametophyte plant. Don't get scared if you see mega gametophyte. Again, it's just saying female, right? But female gametophyte is fine. Inside the megaspore is a female gametophyte. She's starting to poke out just a little bit, but she stays inside the spore. On the female gametophyte plant that is inside that megaspore, there will be archegonia. And in each archegonium is an egg, right? These are things we are getting used to now. Inside the microspore is that male gametophyte plant. Now the male gametophyte plant is just a single antheridium. That's it. That's all it's left. That antheridium is going to have tissue that makes sperm. Here you go. Eventually, the sperm will be made inside of that microspore and be released. And what actually will happen is microspores will land on the megaspores, right? So like that's how the sperm is going to get to the egg in this case. One of these microspores will actually land right onto the female gametophyte that's just poking out a little bit of that megaspore. And so there you go. Here's sperm, and sperm eventually is going to fuse with that egg, right? Okay. Clear drawings. And just so that when you're looking at these later by yourself, same thing I was saying before. Note that the sporophytes now have produced two types of spores, or we can say that they are heterosporous sporophytes. Hetero meaning to different, in this case two, and sporous referring to spores. When you have sporophyte plants that make two types of spores, the gametophyte plants always are going to grow inside the spore. Inside the megaspore, you'll have the female gametophyte. Inside of the microspore, you have the male gametophyte. And because of this, we can say that the gametophytes are dioecious or unisexual in that they only have one type of sex organ on them. So sperm and egg have come together here, right? Here you go. I'll do this golden, right? So like here's your egg and here's your sperm coming to join her. And when that happens via fertilization, you get your 2N zygote, haploid egg, haploid sperm, 2N zygote, right? The beginning of the sporophyte plant, notice the color change. Zygote's going to grow via into that young embryo. And that young embryo will continue to grow, and eventually emerge from where the egg was, because that's where, remember, egg stays. Don't worry about things here. Just know that ultimately that young embryo will start to emerge out of the megaspore, which is where the female gametophyte was. And that brings us back to the beginning. Here's what it looks like in real life. So this is a new sporophyte plant. Here's the stem of the sporophyte plant. Here's its first little baby leaves. There's no seeds here, so we can't call these cotyledons. These are just baby leaves. Because remember, these are spore shedding plants, not seed. There's no seeds yet. This is the root of the sporophyte plant. And this here is what remains of that megaspore, and the female gametophyte is inside of that. But that's eventually this eventually will just fall off, and the sporophyte plant will go on its merry way and do its thing. As shown here. This is where we started in this. Okay. So that's the lycopods. If you want, maybe take a break, you know, take a breather, kind of digest it. I'm going to keep going. That's the beauty of these pre-recorded lectures. And now we're going to switch gears, and we're going to still talk about spore shedding vascular plants, but we're going to talk about ferns, which I love. I love ferns. So ferns, again, you guys don't need to know the phylum name, but you will see it in the book and on some of these slides. So I'm just letting you know. But so ferns are in the phylum Polypodiophyta, and the reason why is because poly means many, and podi refers to feet. And the stems of ferns sometimes look like little rabbit's feet. They're like fuzzy little feet looking things, which we'll see coming up. There's about 11,000 species known. A ton of variation. They're all different sizes. They live in all different places. They have different forms. So we'll see a few of those right now, just so you get a sense of that. So some ferns are super, super small. Here we see two ferns, and that they're on somebody's finger to give you an idea of how big they are. And the one on the left is probably a single plant. The one on the right, the Salvinia, is probably a cluster of about 4 or 5 plants. So just to show you, they can be about a few millimeters to a few centimeters in size when mature. But they can also be trees, depending on the species. So there's a few species of tree ferns, or a few genera of tree ferns, where these ferns actually get over 60 feet tall. Sometimes they grow as epiphytes, which we've come across before, meaning that they grow on other plants, right? So that's what we see here. There's different aquatic species. that we won't really go into the aquatic species in this course. We're going to focus on the ferns that live on land, but there are quite a few of aquatic species that live either submerged or in water or floating in water. They can be small herbs, which is kind of like the ones, like that one on the top left is kind of similar to this one right here. So really little small herbs. And if you're walking around the woods of New Jersey or Connecticut or New York, these are the type of ferns you're probably seeing, right? They're just kind of on the understory of the tree canopy, and they're kind of all around. It's a good time of year to see ferns, by the way. As I mentioned, they can be tree-like, right? Like very tall. If you have ever gone to the New York Botanical Garden in the Bronx, which if you haven't, I really recommend. It's quite lovely, especially this time of year. If you go into the conservatory, the big glass house, they have a ton of tree ferns in there that you can see. Really cool. Where do ferns live? Lots of places. So a lot of ferns live in tropical environments, as shown here. Of course, in our region, which is a temperate region, they occur here as well. They're very important economically as houseplants and landscape plants. Hence, I have several ferns as houseplants in my house because you can often find them for sale as a houseplant. Then if you have a houseplant that's a fern and you'd want advice about it, feel free to take a photo and send me an email. And if anything isn't going good with it, I'm happy to help you figure out what's going wrong. This is what I was saying before about that polypody, right? Like the many-footed plant. So ferns have these stems called rhizomes, right? Horizontal stems. And in some species, the horizontal stems are very fuzzy. For example, here. And so to whoever described the species, the phylum, they thought that those looked like a bunch of little like feet, right? And so that's what the stems look like. And those stems could be above ground or below ground. So the one on the left has stems that grow above the ground. And the one on the right is showing an example of a stem that grows below the ground. We saw this slide before when we learned about leaves. And so ferns and all the other plants that we look at going forward have megaphyls. Now, this is another case of where it's slightly, we have to keep organized because remember, here, and I will do this. This might help. Okay, let's see where to put this. So we saw before microphyll, which is a simple leaf with one vein. And now we're seeing, let's see if I can kind of move this. Can I? Yeah, here we go. Here we go. Microphyll, simple leaf with one vein. Megaphyll is not, it's not necessarily simple. Could be compound too. So we'll just say a leaf with more than one vein. So in Lycopodium, in the Lycopods, which is Lycopodium and Celogenella, they have microfills. They only have leaves with one vein. In ferns and all the other things we're going to look at today and next class, everything else has megaphyll. Megaphyll is a leaf with more than one vein. Don't get that confused with, for example, Microsporophil, which is a sporophyll, aka microphyll with sporangia on it that has microsporangia, right? This microsporangia. You could even do this. Microsporangia on it. And Megasporophil, which is a sporophil. aka, oops, sorry, aka microphyll with Megaspore on it. The reason I'm pointing this out is because, so for these two here, microphyll and megaphyll, that's the only time where micro and mega does not refer to male and female. That's the only time. Every other time you see micro and mega, it refers to male and female. So that's why I'm pointing that out. It can be a little confusing. So rewind that if you need to. Okay, come on, get out of here. Yeah, so ferns and everything else we're going to look at has megaphyll, so leaves with more than one vein. And there's a ton of different forms as shown below. This is just a few examples. Here is our fern leaves, right? You can see they have many veins. Many veins. I'll use yellow, right? In this case, it's a compound leaf. So this is a vein, this is a vein, this is a vein, and there's tiny veins coming out all around. Up here, it's very easy to see, right? Each of these black lines, including the small net-like ones, are veins. And remember, we're in the spore shedding plant, so there's no dicots and monocots. That is only in the angiosperm, so keep that in mind too. And you can see here, each of these white things down here is a vein. So many veins, more than one vein, megaphyll. This we saw during the leaf lecture, right? The parts of a fern leaf. So you have the whole leaf. The whole leaf is like this. Can also be called a frond. And on that leaf, you have parts. You have the petiole, which is the stem of the leaf. You have the green part, which is the blade or lamina. And then the part of the petiole that extends into the blade and the lamina is what's known as the rachis. We've covered this in the leaf lecture already. Most ferns have compound leaves. Some have simple leaves, but most ferns have compound leaves. So just to kind of test yourself, right? You can even take a pause here and just think about it. But which of the leaves above are compound leaves? And I'll let you pause to kind of sort that out. So hopefully you are able to determine that all these leaves are compound leaves, whereas this one is a simple leaf. If you don't know why, go back to your notes in the leaf lecture and things like that. And then also recall these other terms from the leaf lecture, right? Pinna, pinnule, 1 times pinnately compound, 2 times pinnately compound, palmately compound, etc. So if those terms are not familiar to you, go back to that leaf lecture. So when fern leaves come out, they always emerge as this thing called a fiddlehead. And ferns are the only types of plants that have fiddleheads. So if you ever see a fiddlehead, which is this, kind of like a curly Q, right? If you ever see leaves emerging like that, you know you're dealing with a fern. Look, here's some fiddleheads on a tree fern. Look, here's some fiddleheads emerging from soil. Some fiddleheads are actually edible and people eat them. I've actually never eaten fiddleheads yet, which is crazy. I need to do that. People have told me they kind of taste a little bit like asparagus or something like that. I am going to pause to pull up this video. And so hold on, and I will pause recording. Okay, so let me share this video with you guys, which is here. And I'll make it big. Oops, and I'll make it even bigger than that. Oh boy. Big, and make it even bigger than that. So this is fern leaves or fronds emerging from the soil. And I just told you that they emerge as what's known as a fiddlehead. And so we're going to see fiddleheads here. So check it out. Look. So only ferns have fiddleheads. They're the only plants that do this. And you can see there, this is a compound leaf, so you can see the pinna and the pinnules coming out. There's that nodding behavior that we learned about before, right? You can see the leaflets or the pinna starting to unfurl and the little tiny pinnules on each one. Super cool until it finally ends up looking like a fern, a finished fern leaf. So only ferns emerge as fiddleheads. Nothing else does that. Everything else doesn't have fiddleheads. So that's an easy way to remember. If you ever see something that says fiddlehead, you know that you're talking about fern. So that's a good way to stay organized. Okay. So let's keep going. So I said at the top of the ferns that we're only going to talk about the terrestrial ferns, or the ferns that grow on land. We're not going to talk about the aquatic ferns. But just so you know, so the ones that grow on land are Homosporous. The ferns that grow on land only make one type of spore. But the ferns that grow in water are actually heterosporous. That's why we're not going to talk about them in this class. Too complicated. So for the purposes of this class, ferns make one type of spore, because we're only talking about terrestrial ones. And where do they make those spores? So we know, because we're in the vascular plants that are spore shedding vascular plants, that the sporangia are going to be located on leaves. And we know, because we're in the ferns, that the ferns have megaphyll leaves, or leaves with many veins. So sporangia are going to be on megaphyls. And in the case of ferns, the sporangia are going to be located on the bottom side of the leaf, And sometimes the sporangia are arranged in clusters. And when they are arranged in clusters, those clusters are called sori. Is the plural, singular sorus. So this is 1 sorus. This is 5 sori, like sori, not sori, right? Sometimes sori can be in shape of like little round balls. Sometimes a sori will be in lines. Here's another example here where the sori are kind of orangey in appearance. So in each of these, here, I'll just circle this one. That thing I just circled, that yellow thing, is a cluster of sporangia. So each of those little tiny yellow dots that are inside of there here, 1, 2, 3, 4, 5, all, whatever, etc. Each of those is a sporangia, okay? And then a cluster of those sporangia are called sorus, or plural sori. Not all fern species have sori, but a lot of them do. So if you were to look closely at one sporangia, right? So where are we looking here? Just so we get oriented. Look, so okay, it's the same one again. So this is a cluster of sporangia. That's many sporangia. And let's say we're going to look at that one. Close up. Here's what they look like. And fern sporangia are super cool. As I mentioned, they're located on the bottom side of leaves. One way you could remember that is if you see, you'll sometimes see this word, abaxial. Ab means bottom, bottom of the leaves. That's how I remember it. But bottom of the leaves is fine. The reason why that's important to point out is because on Lycopodium and Celogenella, the Lycopods, the sporangia were located on the top side of those Sporophylls. Now we're in the ferns, we have Megafils, and they are located, the sporangia are located on the bottom side of those leaves. Here is what one looks like. You can see inside of this fern sporangia, there's already a bunch of spores ready to be let out. Okay. And what's super cool about fern sporangia is that they act like a catapult. So I don't know if you ever watched anything like Game of Thrones or anything like Vikings, or where people use catapults. Like, so catapult is like this usually wooden thing that you put like a big rock or something, and then you pull it back, and then you like release it, and it shoots the big rock out. So ferns actually, their sporangia does that. It actually shoots like a catapult. And the reason it can do that is because of this special structure of cells. It's called an annulus. So I'll use black. So you see here, this is one sporangia. There's spores inside, and it has special cells that make up the structure called an annulus. And what happens is when the humidity changes, the cells of the annulus are actually sensitive to those changes in humidity, and they change in the humidity causes them to change their shape a little bit, and that causes the cells of the annulus to like open up the sporangia. And so you can see that's what's happening here. And then eventually the tension gets so high that it, boom, shoots the spores out like a catapult. So like plants move and ferns have catapults on them. So there you go. In addition to shooting spores, which is super cool, some species of fern also have a membrane that protects the sporangia so that the humidity doesn't change too much. So this special membrane is called, one of them is called an indusium, and many of them are called indusia. So I'll change colors so it pops a little better. I guess I'll use white. On the left here, we see the underside of a fern leaf with lots and lots of sori, right? Here are 4 sori, right? Clusters of sporangia. Here is one sorus, right? A cluster of sporangia. And you can see the sporangia that are kind of poking out. They look like little quinoa, or like they're little dots, right? Here's some sporangia poking out here for this one. And you might notice that there's this flat structure on top, and that is your indusia. So let me hold on. Annotate. Yes, I guess I'll use dark blue. So do you see this flat thing on top of this sorus? Here's another flat thing on top of that sorus. That's that special membrane called an indusium. On the right here, look, each fern leaflet, so this leaflet has on it 8 sori, and you can see these sori are covered by an indusium that almost looks like a little hamburger bun. And so what the indusia do is they protect the sporangia from drastic changes in humidity, because they're very sensitive to humidity. Here is a cross section through a leaf. And so this whole thing is one sorus, just like this whole thing right here is one sorus, right? A cluster of sporangia. This whole thing is one sorus. Notice it's on the bottom side of the leaf, right? Leaf. And the sorus is composed of many sporangia. There's one, there's one, there's one, etc. And this species has a indusium, which is this little membrane that serves to protect them. It almost looks like a little upside down umbrella, right? You can think of it that way. Like here's the stick of the umbrella and here's like the wide part of the umbrella. But its job is to protect all of these guys, right? It's trying to protect the sporangia. Look, a couple of these sporangia, you could actually see the annulus very well. So like here, you can see the annulus very well. And on that first one I drew too, you can see those special cells that are going to let the sporangia shoot like a catapult. Some species don't have indusium. Instead, they've evolved to have like a false indusium, which is just when the edges of a leaf kind of fold over and protect the sporangia. And that's what's showing you here. So this is not a true indusium. This is a false indusium because it's just the edges of the leaf that are curling over, but they're curling over the sporangia. So they're still serving the same purpose by keeping them from drastic changes in humidity. And these things like the color of the indusia, the color of the sporangia, the shape and arrangement of the sori, it really depends on the species. There's a lot of variety. This is a diagram from your book. But when humidity is right, when the humidity conditions are right for that species, the sporangia are going to shoot their spores. And so, of course, I have videos of that, and I'm going to pause real quick here. So pull them up. Give me 1 second. I will pause this recording. All right, so I've pulled up the videos, and let's share this one, and I'll make it big. So here we're going to see fern sporangia shooting their spores like catapults. All right, so I'll just pause real quick. So this here is one sorus, and on this side we see two sori, right? And this species happens to have an indusium. Here it is. That was covering those sporangia, but the conditions are right now. And then each of these is a sporangium. So all of these together are sporangia. Notice you can see the annulus, right, which is that there pretty well. But anyways, yeah, so these sporangia are going to shoot their spores. So let's let them do it. See, look, the annulus is changing due to changes in humidity. And look at the one on the top. Oh, boom. Boom. Like little catapults. Look at the one on the bottom left over here. Boom. Look at the one up here. Oh. Here we go. Here's some more. So these are fern sporangia shooting their spores. Very cool. I have another one here. Which I will put right here. And pause that for a second, go back to you guys and go to share my screen. This is a different video showing same thing, but in a different species. In this case, the sori are more in like a long banana slug shape. Same idea. I'm gonna fast forward a little here. So each of these things, oh, here we kind of went too fast. So each of these things is a sorus. Look, here, I'm going to pause real quick. Each banana slug looking thing is a sorus, right? It says many sori. And this species has an indusium that almost looks like a banana slug on top, like that uniform thing. And you can see here, this is a sporangia that's starting to poke out. There's a bunch more sporangia poking out under the indusium there. So here you go. Look, that one just shot its spores. Now we're going to get closer. The indusium is kind of pulled back, and you can see all of those little things that are moving are sporangia, shooting their spores. And look at the spores all over the place because they've already shot so many of them. Look, here's a close-up. Boom. Catapult. Boom. Look. Oh, catapult. Boom. So, yeah, gets even closer. That shows the annulus very clearly. Here's a sporangium that's starting to go. It's going to shoot the spores. Boom. Yeah, so you get the idea. The links will be in the PowerPoint if you want to watch more about that. And if you're interested in the physics of how this works, there is a link here, the one on the bottom, that teaches you more about how the fern catapult works, but you don't have to watch that. Just there if you're interested. Okay, so we learned that the fern sporophyte is homosporous. It only produces one type of spore. We learned that the fern sporophyte has megaphyls, or leaves with many veins. We learned that the fern sporophyte has the sporangia located on the bottom side of the megaphils, and that in some species, those sporangia are arranged in clusters called sori, and that they can shoot the sporangia can shoot their spores out like a catapult. And sometimes the sporangia are protected by a layer called an indusium, which gives a little bit of protection. Now I'll talk about the gametophyte. And good for you guys is that we already have talked about the gametophyte generation in ferns, because when we learned about alternation of generations, we used ferns as the example that we went through it with. So fern gametophyte plant is simple thallus. Remember, thallus just means flat green thing. Fern gametophyte is in the shape of a heart. It's a little green heart. I know it looked kind of blue on this slide. And because it's green, photosynthetic, and it has these little hair-like things again, right, rhizoids, that help anchor it to wherever it's going to be growing. Because it's a gametophyte plant, and because they're homosporous, right, the sporophytes are homosporous, produce only one type of spore. We covered today that if there is only one type of spore, then the gametophyte that grows from the spore will, number one, grow outside of the spore wall, but number two, have both male and female sex organs located on it, right? It'll be monoecious. So here you go. Female sex organs toward the center of the fern gametophyte. Male sex organs are kind of like spread around. Fern gametophytes are super, super, super small, super small. So, and this is a person holding, circled in orange is the fern gametophyte plant, and the new sporophyte plant is already emerging out of it. So fern gametophyte plants are super, super small. This is what we saw in the last class, where we learned about alternation of generations using a fern. Today, we're going to go in through the fern. It's a little bit more complex than what we saw that first class, but it's the same players. So here on the left, fern sporophyte plant, I'll use blue. Right, adult, oops, that's not blue. Adult sporophyte plant, 2N, fern. Same players as before, horizontal stem is a rhizome, roots coming off of the stem, adventitious roots, right, coming off the stem. And then we have our leaves, right? Our leaves are a megaphyll, right? A leaf with more than one vein. On the bottom of those leaves are clusters of sporangia called sori. You can see them here. Each of those dots is a sorus, one sorus, many sori. Now we look closely, look, we're going to look right there. And that's what this is showing us here, close up. So here's one sorus, or cluster of sporangia. And we're going to look at one of those sporangia, this one. They're going to make it big. Inside of that sporangia is tissue that is 2N that's going to undergo what? Meiosis to produce spores. Purpose of sporangia is to make spores. That's its job. And it can't make those spores without meiosis because it has to go from diploid to haploid. Okay. I'm going to move my head here. So if my head's blocking you here, you pull up the lecture slides that are available on Canvas to see this part. But basically, there is an annulus, and we learned about that already. It's that cell layer that is sensitive to humidity, and eventually the annulus is going to pull back and then shoot out the haploid spores, which is what's happening probably behind my head. So please pull up those slides on Canvas. Here, oh, here it is here. So you can see here, here's the cells of the annulus. You can see a bunch of spores being ready. Here, I'll use a different color because the spores are haploid. And I move this thing too many things in my way here. Look, spores are haploid, and we're going to talk about just one spore, right? So I'm going to track this one spore here. Gametophyte plant, remember, there's only one type of spore. So gametophyte plant grows outside of it and has both sex organs on that gametophyte plant. So here we go. Gametophyte plant is emerging outside of the spore. Here it is, finished, heart-shaped, photosynthetic, both male and female sex organs on it, right? Female sex organs, male sex organs. And right, and as I said, we're only looking at the terrestrial sporophytes, the terrestrial ferns, and so fern species for the case of our class have one type of spore, gametophytes that grow outside of the spore. Eventually, this should be coming quicker now because we've covered it so many times. Eventually, the sperm will be made in the antheridium. Egg will be inside of that archegonium. Eventually, the sperm and egg are going to fuse. Look, here, sperm is swimming to the egg. And when that happens, the process of fertilization, sperm, haploid egg, diploid, zygote. Right there. Okay. The diploid zygote is going to grow via what? Mitosis. Grow via mitosis into the young embryo. The young embryo is going to grow via mitosis into mature sporophyte plant. And eventually, so like here is that gametophyte plant where the egg was and where the sperm came from in this case. Egg and sperm already fused together, right? Because we have that diploid zygote and then embryo. And so the new sporophyte plant, which I'm drawing in blue, will grow out of that gametophyte plant, and then eventually the gametophyte plant will just disintegrate, and the new mature sporophyte plant will go on its own independently because it's the dominant generation. And as a reminder, in all the vascular plants, the dominant generation is the sporophyte. And in all the non-vascular plants, the dominant generation is the gametophyte. We talked about that at the top of the class. Okay, oh yeah, notice, new leaves emerging as a what? Fiddlehead, right? Because only ferns have fiddleheads. Okay, maybe take a break. I know this is a lot of material. This is why we're doing it pre-recorded. Now we're going to go talking about the seed-shedding vascular plants. I will also take a little break and refresh my tea. So see you in a second. Okay, I opted for water and seltzer instead of tea. Too much tea. Okay, let's get back to it. I think you guys can see this, but let me just make sure, because that always won't be good. Okay. All right, yes, I see this and I see myself and it says I'm recording, so I think I'm good to go. All right, so far in the course, we've covered the non-vascular plants and we've covered some of the vascular plants, which were the spore-shedding plants, right? Lycopods with Lycopodium and Celogenella, and the ferns. So now we're going to go into the seed-shedding vascular plants, the seed-shedding plants, which are the majority of plants. The seed is a big deal. The seed is a big deal, not just for humans, but for the plants as well. It's a significant evolutionary milestone for plants, the ovule and the seed. And these are the plants that make up most of the plants on Earth, are the seed-shedding plants. And that's part of the reason that is because it is such a successful way for plants to exist. The seed and the ovule work way better than just releasing spores. Releasing spores is fine, but it's not as good as the seed. So how are seeds evolutionarily advantageous? Say that 5 times fast. Well, seeds provide great survival value to the developing embryos. So embryos are developing inside of a seed, and that gives them better protection. And then the seed also provides a food source for the embryo, which spores don't do anything like that. So this gives plants that shed seeds an advantage over plants that just shed spores. And we can tell that advantage because of how many plants on Earth actually make seeds. It's the better way. So don't worry, don't get nervous when you see this. You don't have to know the phylum names. It's just, it was easier. This was a photo from a previous lecture I did. But I don't want you to get scared here. So here's what I'll do. So mosses, liverworts, here, mosses, liverworts, hornworts. Look, mosses are Homosporous, liverworts are Homosporous, hornworts are Homosporous, right? Let's put another one of these text boxes here. Oops. And then, just so you don't get scared. Well, those two are fine. Lycopodium, Homosporous. Celogenella, Heterosporous. Horsetails we didn't cover, but they're Homosporous. And then these, don't worry about these here. The ones I'm covering over are those aquatic ferns, right? But look, so you have from the top, mosses, liverworts, and hornworts, all Homosporous, one type of spore. Sporophyte makes one type of spore. Lycopodium, Sporophyte makes one type of spore. Celogenella, Sporophyte makes two types of spores, right? Microspores and Megaspores. Horsetails we didn't cover, don't worry about, Ecocetophyta. And then ferns, the terrestrial ferns, are also Homosporous, right? That's what we see here. Don't worry about those ones on the bottom. Oh gosh, gotta get it out of the text box thing. Then we get to the seed shedding plants, which we're going to talk about today. We'll start with the gymnosperms. And look, seed shedding plants always make two types of spores. Two types of spores. That's what I'm trying to get you to see with this thing. All seed plants are heterosporous. They have megaspores and microspores. Megaspores are going to give rise to that female gametophyte plant, and microspores are going to give rise to that male gametophyte plant, just like we saw in Celogenella. However, in the seed plants, we have this like extreme form of heterospory in that we have the ovule, and the ovule is going to develop into the seed. We may have talked about it briefly during the flower and germination lab lecture, rather. But just so you know, an ovule is a structure in seed plants that contains the female gametophyte with egg, and therefore the gametophyte, female gametophyte, is better with her egg, is better protected by the layers of the ovule, which are called the nucellus and the integument, which we'll see on slides coming up. After the egg is fertilized by sperm, so egg is in the ovule. After egg is fertilized by sperm, the ovule will turn into a seed. And inside of that seed will be the embryo, or the zygote, which grows into an embryo. And that's because egg and sperm have united. And so now we have a seed instead of an ovule. This is an ovule. This is some basic anatomy of the ovule. So you have megasporangium, which I'll use blue. So you have megasporangium, aka nucellus. They're the same thing. Megasporangium is fine. And that's this whole thing here. And inside of that is where your megaspore is going to be located. Then you have, and then megaspore is inside of the megasporangium. So the megaspore no longer gets released from the megasporangium, whereas in Celogenella it did. In Celogenella, the megaspore left. Now the megaspore stays inside of that megasporangium, never leaves. And the megaspore is going to grow into that female gametophyte plant, and the female gametophyte plant is going to stay in there as well. Surrounding that are these layers of tissue. In some seed shedding plants, they have one layer. In other seed shedding plants, they have two layers of integument. That's how you say that, integument. And that's just a layer of tissue that surrounds the megasporangium and therefore the female gametophyte when she grows in there. And then after fertilization, the integument is what's going to be turns into the seed coat, which is a part of a seed. And then lastly, micropyle. Remember, this is parts of an ovule. Micropyle is this opening up here. That's where sperm is going to be able to enter to meet the egg when there is an egg in there. Right now, in this drawing, there's no egg in there yet because the female gametophyte has not grown yet from the megaspore. Okay. After the egg is fertilized, the embryo is going to grow inside of that female gametophyte plant. So inside of that megasporangium, and this is what we saw during that seed lecture, right? Yes. This whole thing in the middle is your new sporophyte plant, surrounded by the female gametophyte plant because that's where the egg was. And the female gametophyte plant was inside of the megasporangium, which is what this is showing you. And further, that's inside of those integuments. So it's like many, many layers protecting the egg and then ultimately protecting the embryo once the ovule turns into a seed. So as I said, majority of plants on Earth are seed plants. So this pie chart shows you all the different types of plants on Earth, and green represents the seed shedding plants. So this is how we know that ovule and seed give these plants an evolutionary advantage, because otherwise there wouldn't be this many of them that have seeds. So here are the seed shedding plants. You have gymnosperms, which we're going to cover today, and then angiosperms, which we'll cover during the last lecture. Of those seed shedding plants, so if we were to take the green part of this pie chart and make its own pie chart with just the green part, this is what it would look like. The majority of the seed shedding plants are flowering plants, about 370,000. And then there's about 11,000 that are gymnosperms. So angiosperms are flowering plants, gymnosperms are not, but they still make seeds. So here's our roadmap, and today we're going to cover those gymnosperms. So, as I said, flowering plants are called angiosperms. Before we can learn about the gymnosperms, we have to get a couple of terms down. So gymnosperms comes from naked sperm, which in the case of the Greek, sperm refers to seed. So the word gymnosperms means naked seed. And what that means is that naked seed. What does that mean? What that means is that the ovules, and after they're fertilized, the seeds, right? So ovules, once ovules are fertilized, they turn into a seed. But the ovules, and then later the seeds, are not enclosed in an ovary. They are not enclosed in an ovary. They are visible with the naked eye and exposed to the environment. So that's what gymnosperm means, naked seed. So it's naked ovule, because the ovule is not, the ovule is not enclosed in an ovary. So naked ovule, and then ultimately naked seed. Whereas in the angiosperms, angion means a vessel or a container, and seed, sperma still refers to seed. So you have a seed in a vessel or a container. So in the angiosperms, which we'll learn about in the last class, the ovules, and later the seeds, once the ovules are fertilized, are enclosed in an ovary, so they are not exposed to the environment. That's the difference between those two things. Today we're going to learn about gymnosperms, which have ovules that are not enclosed in an ovary. They are visible with the naked eye, exposed to the environment. Here are some gymnosperm ovules. Notice they're not in a flower or anything like that. They're just out there. So here's an ovule on a leaf. Believe it or not, these are also ovules that are just on a weird looking leaf. Here's an ovule. And these are also ovules that once this cone is finished, they will be exposed. So they are exposed and visible to the naked eye. here is a diagram just demonstrating what I explained. On the left, you have gymnosperm. And look, here's an ovule. See, micropyle, integument, same thing we just saw, not enclosed in anything. Whereas over here, here's an ovule of an angiosperm, and it's surrounded by an ovary. So naked seed over here, right? Gymnosperm, which is what we're going to cover today. So that should be pretty straightforward, I think. All right, the gymnosperms we're going to learn about are the cycads, ginkgo, and conifers. And for some of these, we're not going to go into detail that we've gone into so far because we're getting to know the players. So we can go through them a little quicker. So first we'll talk about cycads. Cycads look like palm trees. They look like palm trees, but they're not. But they do look like them visually. And that's because the word cycad actually comes from a Greek word that does mean palm, because they do look like palm trees, but they're not palm trees. Palm trees evolved millions of years later. There's a lot of there's not that many species left on Earth. There's about 300 species. Most of those are facing extinction for different reasons, including slow reproduction, habitat destruction. Sometimes government remove them because they're poisonous to certain livestock, and also seed predation, such that the seeds get destroyed and can't grow into another plant. Cycads are old. They evolved around, they first showed up based on the fossil record, they first showed up during the Permian, but then during the Jurassic period, like Jurassic Park, right? During the Jurassic period, there was a huge explosion in the number of cycad species, so a lot of diversification. And what that means is that cycads are plants the dinosaurs ate. Yes, which is super cool to think about that. And also, I cannot resist showing these old diagrams, these old drawings, because they're just hilarious. Like, look at this little dinosaur eating the cycad. Like, oh yeah, like it tickles me. But yeah, a lot of times when you see drawings or reproductions of, you know, the Jurassic period, a lot of the plants you'll see in the background are ferns, but also cycads, and cycads make seeds, and so dinosaurs would have eaten not only the leaves, but also the seeds. Yeah. Plants the dinosaurs ate. Cool. Yeah, so cycads used to be worldwide, but now they only occur in tropical and subtropical regions. So parts of Africa, the northern part of South America and most of Central America, Southeast Asia, the South Pacific, and Australia. And there's even one species that occurs naturally in Florida of the United States. So here's our ovule again. We already saw this a couple slides back. Just a reminder that inside of the ovule is that megasporangium, and inside of that megasporangium is a megaspore, one megaspore. For the microsporangia, they're also located on leaves. The leaves don't always look like leaves, but they're located on leaves, and they, just like we have seen before, produce the microspores. So this little yellow Pac-Man thing is one microsporangia on this weird looking leaf. Yes, and so just like we saw before in Celogenella, the ovules with the megasporangia inside and the microsporangia are attached to leaves that are called Megasporophylls and Microsporophylls, just like in Celogenella. In the megaphyls, the megaphyls are sometimes arranged in a strobilus, other times not. It depends on, so remember the strobilus thing, right? Little organized sporophylls, right? Where each leaf has a sporangia on it. Megasporophylls are sometimes arranged in a strobilus, but microsporophylls are always arranged in a strobilus. So that is helpful for identifying. Just so you know, like these are just some representative cycad strobili. So on the left here, both of these are each of these is a strobilus. And this tells you on the slide, right? A strobilus is a stem with leaves, and the leaves have sporangia on them. Because we have two different types of spores, in the case of the cycads, you'll have a strobilus that only makes megaspores and a strobilus that only makes microspores. On the right here is a female strobilus. On the left, next to it, is a male strobilus. Both of them have stem. And these little brown things, believe it or not, are weird leaves. Same goes over here. And the ovules will be located on the leaves over here, whereas the microsporangia will be, they're all the little kind of yellowish, pale colored stuff you see in between the leaves there. There is a lot of diversity in cycad strobilus shape. They could be different colors. They can be different sizes. Depends on the species. So this is that same female strobilus I showed you on the previous slide, right here. And then this is like a drawing kind of it. So each of these, they almost look like oyster crackers, those crackers that you put in soup. Each of those things is a leaf or a megasporophyll. And there is ovules on each one. It's harder to tell over here, but like each of these reddish things, is a leaf. And there's a little reddish ovule right there that's on this on this leaf here. Sometimes the book or other sources will call these cones, cycad cones. No, they're not cones. They're strobilus, or if there's many of them, they are strobili. Cones are only on the conifers, which we'll cover later at the end of this class. Number of ovules on a megasporophyll leaf will depend on the species. The one on the left here has 5. The one in the middle has 6 ovules, and the one on the right has 2. Cycads are strictly dioecious. Dioecious, as you know, means you have plants that have either male or female sex structures on them. So the male plants, the male sporophyte plants, only have male strobili. And here they are. Here's some male strobili. We just looked at the female one more closely. Now we'll look at the male. So this whole thing is a male strobilus. If we look at a drawing of it, it's a stem, right? So there's like a stem in here with all these tiny little leaves on it. And each of those tiny leaves bear sporangia. They bear microsporangia because they're going to make microspores. So if we were to rip one of these tiny leaves off, this is what it looks like. And all these little dots on the bottom side of the leaf are the microsporangia. Here's what it looks like in real. So this is the stem of a strobilus, right? Like if here's the strobilus, it's a stem. Here is a, here is a microsporophyll leaf. Here is a microsporophyll leaf, etc., going all the way around. And on the bottom side of each of those leaves, you see the microsporangia. Lots and lots on each leaf. Strobilus, leaf, aka microsporophyll, right? Okay. In addition to the ovule being the ovule and seed being a big deal in terms of the evolution of plants, the pollen grain is also a big deal in terms of the evolution of plants. So those microsporangia that we just looked at, right? So on the bottom of these leaves, the one leaf is circled in purple, right? A drawing of a leaf is circled in purple, and then a real one is circled in purple. On the bottom side of the leaf, all those little dots are the microsporangia. And inside those microsporangia are those mother cells, right? Those 2N microspore mother cells. And those are going to be the ones, you're in a sporangia, so in a sporangia is where meiosis occurs. And in those microsporangia, you're going to have meiosis occur to produce microspores. The microspores that are made from meiosis inside of that sporangia are going to grow a little bit via mitosis and produce this male gametophyte plant, which is really, really reduced now, and we call it the pollen grain. And I'll show you how that works on the slides coming up. Oh, what is this? I'm gonna move that. Oh, yes. And just like the plants we looked at before, the cycad sperm do swim. They have lots and lots and lots of little tails, which are shown here on the right of this sperm. But they don't swim through water anymore. That's a difference. They now swim down a structure called the pollen tube, which we will learn about in a few slides. And you can see cilia is the little tails. So each sperm has lots and lots of little tails that help swim. Okay. And it can take months between when pollen lands where it needs to land, pollen grain, where it lands on the ovule. It can take months between that event and the sperm from that pollen grain fusing with an egg. And this is part of the reason why cycads are facing extinction, because they have very, very slow reproduction. Eventually, the sperm and egg will fuse, and the result will, the ovule will then turn into a seed. So these are examples of some seeds that are finishing up, and they are on those. They're on the leaves that had the ovules on them, the Megaspores. Eventually, the seed falls off. Inside of the seed is the embryo, right? And so eventually the seed will germinate and the embryo will come out of the seed, and then you'll have your new cycad plant as shown here on the right, which will grow into a new mature sporophyte plant. So that was kind of a crash course through cycads. The main takeaways are that cycads have dioecious sporophytes. So they have sporophytes with male. Some sporophytes only bear male strobili. Some sporophytes only bear female strobili. That's fine for this class. On the strobili are leaves. On the leaves are sporangia. Because there's only male and female ones, the male strobili have microsporangia that make microspores, and those microspores turn into pollen grains. On the female plants, the leaves of the strobilis have megasporangia, which make megaspores, but now those megaspores are inside a structure called an ovule because we're in the seed shedding plants. So the pollen grain is the highly reduced male gametophyte plant, and the ovule contains the very small female gametophyte plant who lives inside the ovule now. All right, so now I'll talk about ginkgo. Ginkgo is a tree, can get rather tall. If you live in New York City or Journal Square or other type city areas, you've probably seen it. Common name of it, so it comes from the phylum Gingophyta, which you don't need to know. It comes from the genus Ginkgo, which you don't need to know. But its common name is ginkgo tree, so you can't get away from learning ginkgo. Sorry. We just call it ginkgo. Interestingly, there is only, so you have this whole phylum of plants, and in that whole phylum of plants, there's only one genus and there's only one species. So there's only one member of ginkgo left, and it's Ginkgo biloba, but ginkgo is fine for us. Very popular in temple gardens, and although it's a very common street tree and planted in gardens, we are not really sure if there's any remaining wild populations, but ginkgo is fine. It's planted all over the world, but don't think there's any wild populations left, just cultivated. Here's what the leaves look like. They kind of look like fans. Here's one leaf. So they are fan-shaped. We say that they're bilobed because they have, oops, I gotta move this thing. Because they have 1, 2 lobes. So this, notice that this leaf has lots of veins in it because it's a megaphyll, right? So That's what ginkgo leaf looks like. It has a very long petiole. Ginkgo, so when we learned about the Lycopods, we said they were the oldest surviving vascular plant. And that's true. But ginkgo is the oldest living or surviving seed plant. So ginkgo dates back to around 350 million years ago based on the fossil record. And at the bottom of this page, you can see a ginkgo fossilized leaf. So this here, this is a fossil of a ginkgo leaf. And this is a modern day ginkgo leaf. So they look very, very similar and have not changed much over those millions and millions of years. And because of that, botanists call the ginkgo species, Ginkgo biloba, the only species, a living fossil, because it still lives today and it looks almost identical to how it looked 300 million years ago. And just so you know, like lobing of the ginkgo leaf, even now when it's developing, it can be more lobed. It becomes bilobed when it's finished. So that's why there's some variation there. As I said, very popular as a street tree, planted all over Brooklyn, all over the Bronx, all over Jersey City, Journal Square. Very, very common all around the world as a street tree. Part of that is because it does so well. It doesn't mind the bad conditions of a city in terms of pollution or lack of soil. It grows just fine. They can get real tall and they can get real wide. So up to 100 feet tall and up to about 4 feet wide. And because you've already learned about stems and you're thinking to yourself, oh, it's a tree and it gets so tall and wide, that must mean, what does that mean about the growth of ginkgo? Because it gets, not only does it get really, really tall, but more importantly, it gets really, really wide. The stem gets super wide. Ding, ding, ding. Thinking about growth, has abundant secondary growth, right? Has abundant secondary growth. Because that's the type of growth you need for widening, right? Look at that. Wow, wide. Same slide we saw actually during that STEM lab. Ginkgo is deciduous. That is a word that means the leaves fall off in the winter. Or actually they fall off in the fall. That's why it's called fall. So yeah, ginkgo is deciduous. The leaves fall off every year. And another reason why it's so popular as a street tree is because ginkgo has this beautiful golden foliage when the leaves start to fall off. See? Another thing ginkgo has is these short shoots and long shoots. I don't really care about this too much, but it does help you identify the tree. So this circle here is showing you an apical bud, which you should know what that is already. The apical buds make those long shoots, which are shown in blue here. Lateral buds remember, lateral buds are for branching, and so the lateral buds make short shoots, which are shown here in red. There's another way to look at them. I don't care about the end nodes and internodes. The short shoots, I like this. The short shoots emerge from the long shoots. The leaves emerge from the short shoots. So yeah, the leaves and the reproductive structures are going to be emerging from those short shoots. What's cool about this is that because of these long and short shoots, even in wintertime when the plants don't have any leaves on them, it's super easy to identify ginkgo. I can see a tree 2 blocks away and know that it's a ginkgo tree because of this peg-like look to the branches, because it makes them very easy to identify in wintertime. Okay, just like cycads, ginkgo is strictly dioecious, so they have male and female plants. The female plants bear only ovules, and the male plants bear male strobili. Yeah. Here is an example of what the female plants will look like. So you have ovules, and importantly, which is different than what we saw before, the ovules here are not in a strobilus. The ovules are paired. Here's two of them, paired and on a stalk. So here is a stalk, and here's one ovule, and here's another ovule. Here's two, right? A pair of them here on a stalk, a pair of them here on a stalk. And those will emerge right next to the regular leaves on those short shoots. So those what the female look like, ovules paired on a stalk. Ovules are paired on a stalk. Make a note of that in ginkgo. Here you go again. So ovules are paired on a stalk. One ovule, two ovules. Notice you can see the ovules with your naked eye because they are gymnosperms. They're not inside of anything. Paired on a stalk, right? See them with the naked eye as they're from gymnosperms. Here is the male strobili that grows on those male plants. They're going to strobili. So here is one strobili. Here's another, or one strobilus. Here's another strobilus. So two strobili. And just like the ovules, they're going to emerge on the short shoots right next to the regular leaves. The parts of the strobili are the same stem with leaves that have microsporangia on them. And so eventually those microsporangia are going to open and release pollen grains into the wind. And then eventually those pollen grains will land on one of these ovules, and that will allow the sperm to eventually reach the egg in the ovule. And compared to cycads, ginkgo, time between pollen landing on an ovule and sperm and egg uniting is a lot quicker in ginkgo compared to the cycads. Eventually, once the sperm from the pollen grain fertilizes the egg in the ovule, you'll end up with a seed, and the seeds will be where those ovules were. So just like you had ovules that were paired and on a stalk, obviously once those ovules become seeds, they will also be a pair of seeds on one stalk. So that's where they were ovules before they were fertilized. Eventually the seeds mature and fall from the tree, and ginkgo seeds stink. They smell awful. And it's funny because this is when you need botanists to help your city planners. There are stretches in the Bronx and Brooklyn where they have only planted female trees. And so when the seeds are ready, all the trees on a whole block will drop these stinky seeds. Smells almost like somebody once told me it smells like, described it as like rotten butter. And I was thinking, like, first of all, how do you even know? I didn't even know butter could go rotten. Like, how do you know what rotten butter smells like? But I was like, okay, yeah, let's just say it smells bad. But if you've ever encountered this before, you know what I'm talking about. It smells really bad. So now that people have taken that into consideration, a lot of times only the male trees are planted. But complications from that as well, because if you're allergic to ginkgo pollen, which is different than your regular pollen. So I don't know to what rate people are, but putting a bunch of male trees all in one block could be a problem too, maybe. So eventually seeds fall off the tree, and that's that. Okay, this is the last part of this lecture. I know this lecture is mad long. That's why we're pre-recording it, so you can take it in bits. The last lecture that we have coming up, right, the last number 8 is not going to be as long. This is the longest one, I think. Plus, I had a tornado, so that kind of threw me for a loop too. All right, so now we're going to talk about the last of these seed-shedding vascular plants that are gymnosperms, the conifers. The conifers, out of the 3 of these, cycads, ginkgo, and conifers, probably the most familiar with conifers, just because of things like the Christmas tree trade. So they're gymnosperms and they're conifers. Conifers make cones. Conifers make cones. Nothing else makes cones. The name even says it, conifer. fir, I don't say that correctly, but fairer, actually, fairer comes from the Latin to carry or bear, and conus, which refers to a cone. So the name conifer means to bear cones. That's what it means. Conifers have cones. Nothing else has cones. Even if the book even puts it in parentheses, it's no such thing, only conifers. And as you can see from this diagram on the bottom, conifer cones come in a lot of different shapes and sizes depending on the species. And I have a few here to show you just to give you an idea. So this little guy here is from a hemlock tree. And look how teeny tiny, look, so tiny, right? Then you have this a little bigger from the Dawn redwood tree. Let's see what else I got here. These are actually from sequoia trees. So sequoias are huge, but their cones are not all that big. Still cool looking though. Slightly bigger here. This is from an Atlas cedar, so slightly bigger. And then this is from a spruce. Ooh, slightly bigger even yet. And then some cones are even like this, and even bigger. There's even bigger ones. So the point is a lot of different shapes and sizes. When they're finished, they tend to be brown and dry. But yeah, just to give you an idea, maybe an excuse to like dig into my specimens that I've collected over the years. So yeah, only conifers have cones. Conifers, they're trees. They're trees, they have a lot of wood, they have a lot of bark, so they have a lot of secondary growth, just like your ginkgo. And the largest trees in the world are conifers, and that's what I was just saying. So look here. This is so the largest trees in the world that live today are conifers. So I'll get you here just so you can get an appreciation for what's going on in this slide. Do you see on the right this dot? That's supposed to be a 6-foot human. I circled it in blue, but here I'll put a line under it. So that dot is supposed to be a 6-foot human. Check out the size of these trees. I mean, these giant sequoias and coastal redwoods, those are the ones I was saying, like the sequoia trees can be that big. Look, 300 feet tall and their cones are this big. Okay. But yeah, Douglas fir, it regularly happens in New Jersey, and it could even reach up to 250 feet tall. Giant trees. Tallest living tree in the world is one of these sequoias, and its name is, they've named it, of course, because it's so big, is Hyperion. It's in California. It's about 400 feet tall. It's one of those coastal, so I should be clear here. The giant sequoia and coastal redwoods are both types of sequoia. So here you go, Sequoia sempervirens, it's that coastal redwood. And just to give you an appreciation, because this is pretty amazing. I haven't visited this one yet. I hope to one day go with, but look, this is a person who's sitting. It's not me. I wish it was. But look, they're sitting. So look at the size of this trunk. I mean, pretty incredible. So yeah, tallest tree in the world is a conifer. Give you an idea of how big Hyperion is. Here is Big Ben from London, and here is our Statue of Liberty. And next to that is Hyperion. So bigger than the Statue of Liberty, kids. I know. What? That's why I want to go see it someday. So super, super big. So conifers are the oldest trees that live today. Don't get that confused with the ginkgo. With the ginkgo, its lineage is the oldest lineage, in that ginkgo trees have existed on Earth for hundreds and hundreds of millions of years. But the Hyperion, oh sorry, that's tallest living tree yet, right? Oldest living tree. So this means like these trees are alive. They're one individual that's been alive, not like a lineage. And so, for example, the bristlecone pines, which are also in California. California's got a lot going on with trees. are the oldest plants, meaning been alive, like one individual being alive. So there's some there that are more than 4,600 years old for one tree individual. And scientists use these trees to better understand the aging process, because we're like, how has this tree been alive for over 4,000 years? That's something we need to know. And then there is this other old tree, which is slightly not like bristlecone pine's pretty impressive because it's one individual who's been alive for over that, almost 5,000 years. These are also super cool, but it's not quite the same thing in that. So these trees, the Norway spruces, are able to make new plants using asexual reproduction, like that we learned about during the non-vascular plant lecture. And so scientists have used DNA to determine that this plant here, which is called Old Chijiko, I think that's how you say that, in Sweden, is a clone of a plant that started almost 10,000 years ago. So like there was a plant almost 10,000 years ago, and it made a clone of itself, and then that plant made a clone of itself, etc., such that we still have this one today, almost 10,000 years old. So that means that the plant that made this vegetative, the lineage of vegetative clones that would have made this plant would have sprouted in 75, 50 BC. And just to give you a perspective of how long ago that is, writing, like human writing, was not invented until around 4000 BC. So impressive. All right, but back to conifers. So there's about 500, 600 species worldwide, and most of them have leaves that stay on year round or are evergreen. And the leaves live for several years and then eventually get replaced. Conifers dominate the northern latitudes. There's these boreal forests that are in the northern part of the globe, and that's where those areas are mostly conifers. Other things can't survive up there. We use conifers for a lot of different things, and because they dominate that northern part of the globe, they actually do remove a lot of CO2 from the atmosphere. For example, the Russian boreal forest is many times larger than the Amazon rainforest. So it's a lot of conifers removing a lot of CO2 from the air. But we also use them for different things like making paper, particularly toilet paper, which is a little weird, lumber, furniture, fuel, and other things. Like we eat pine nuts, and we make jewelry out of this thing called resin, which I think I have a photo coming up. As I said, they dominate northern latitudes, and that's because they have evolved to deal with living in very dry conditions. So a lot of times, this is a new term, xerophytic, just means dry. Plants that can live in dry places. Xerophytic. Oh, I'm using white. That's why you can't see. Xerophytic plants means xeros comes from the Greek. Xeros comes from Greek, which means dry. Xerophytic plant. So conifers live in these very dry environments. For example, sandy soils, as in New Jersey. Like if you go to the Pine Barrens in Jersey, it's very sandy soils there. But also, if you're living in a place where water is frozen, it's basically a dry environment. Like if the water is frozen and you can't access it, it's dry. So they're adapted to these environments, and the way they have adapted to that is by their leaf shape. So some conifers have these needle-like leaves, which don't have a lot of surface area, so it helps them deal with the cold. Scale-like leaves, awl-like leaves, which are named after this old leather working tool called an awl-like leaf. Some of them have more laminar leaves, or like more like leaves we're used to, where they're flatter and greener and maybe a little broad. But part of the reason why conifers can live in these dry environments is because they have evolved sunken stomata, and that helps them reduce how much water vapor is lost through their leaves. So here's a cross section through a conifer leaf. And these circles are showing you stomata that are sunken. So for example, this is one here. So they're sunken into the tissue of the leaf, which makes them less exposed to air, which makes them lose water vapor less. In addition to the sunken stomata, conifer leaves also have a thick cuticle, which we learned about in the leaf lab, and that overall reduced surface area, because like a needle has a lot less surface area than like a broad leaf, right? You don't need to know terms on this. I just want to show you a different way of looking at the stomata. It shows you a stoma down there, so it's sunken. Another thing that conifer leaves have, is resin ducts. When you think of like a Christmas tree or like a pine tree, you think of that smell, like that smell of that tree, and that's because of resin. The resin is what gives it that smell. And in addition to some people enjoying that smell also, or that resin also helps prevent the trees from being infected by fungus, and it makes insects not want to eat the tree because it doesn't taste good, which helps protect the trees. In addition, that resin is why we have these things called amber fossils. So again, back to your Jurassic Park, I don't know if you've seen that movie, but like there's a whole scene of like, you know, there's a mosquito in a piece of amber and they use that to get the DNA. Yeah. But the point is amber. Amber is a thing. It's basically fossilized resin from pine trees, or from conifers. And sometimes small animals can be trapped. So back in evolutionary geological time, a small animal would have gotten trapped in the resin. Then the resin fossilized over millions of years, and so we still see the animal inside, which is kind of cool. People also use this to make jewelry. Conifers are usually monoecious, such that one sporophyte plant will bear the male and female reproductive structures on it. In the case of conifers, the male reproductive structures are strobily, just like we saw in ginkgo, just like we saw in cycads. Whereas the female reproductive structures are going to be on cones. So the ovules and then later the seeds will be on cones. So there you go. Male reproductive structures are on the strobily shown on the bottom. Female reproductive structures are on the cones. The strobily, just like we have already seen, have on them microsporophylls that have microsporangia on them that are going to make the microspores that make the pollen grains. So eventually the pollen grain is going to get released from the strobily, as you can see here on the right, and be put into the wind. Female reproductive structures, so the ovules are on cones, and then once they become fertilized, the seeds are also on the cones. As I said, the pollen is brought to the ovule via wind. It's the primary method of seed dispersal as well. So wind makes the pollen go to the ovule. Wind also makes the seeds leave the cones. So on the left here, you see pollen leaving the strobily. And on the right here, you see a pollen grain. So that is a conifer pollen grain. And notice they have these little air bladders. I'll use black. So this is a pollen grain. And then this is a little air bladder. And this is a little air bladder, whoops, coming off of it that helps it fly in the wind and also helps orient it to touch the ovule. And one way that people always remember how this is like conifer pollen, you have the pollen grain and you have the little air bladders on top. So it kind of maybe looks like somebody we've seen before a little bit. Okay. That's how some of my students remember it and memorized it in the past. I mean, look, oops, look. I mean, I can see it. I can see it. Look, a little pollen grain here. Got the little air bladder here, little air bladder here. Look, you can even see cells inside of the pollen grain. Kind of like a, oh my God. Okay. I'm losing it, kids. All right. So pollen grain under the microscope. Yes, that's what we see here. Air bladders, and inside the pollen grain are some cells. You have a tube cell, which is shown there, and then some other cells. These are seeds, and the seeds also get dispersed by wind, and they have a little wing on them that helps with that. So the pollen grains have an air bladders, and here is a seed. So this would have been an ovule that has now been fertilized. And when it's ready, it actually has a little wing, which helps it fly away from the tree. Okay. Some species are dispersed by birds because they have a fleshy cone. So it's not your standard, like, dry looking cone, but instead it's more fleshy. And birds will eat those and poop them out, and the seed will germinate somewhere else far away. And then this is our alternation of generations with conifers. So I'm going to try to go through this relatively quickly because a lot of the players should be familiar by now. So you have one tree with both male and female reproductive structures on them. On the left, you see a Strobilia. On the right, you see cones in these. So here is Strobilia. Here is a cone. Here is a cone. Strobilia, just like all the Strobilia we've seen before, have a microsporophil and microsporangia. Inside the microsporangia are those spore mother cells that are 2N, and those are going to be what undergoes meiosis to make microspores. On the cones, you have ovules, and in each ovule, you have Megaspore mother cell, which is 2N. And Megaspore mother cell is going to be what undergoes meiosis to make the Megaspore, which will grow into the female gametophyte. So here's your male Strobilus. This is just showing you one. So you have a stem in the middle. You have, I'll do this here. Here's a leaf. Here's a leaf, aka microsporophyll. On the bottom of each leaf is a sporangia, aka microsporangium, because it's making microspores. Okay. Here is a cone cross section. I think I do it for you. So you have a cone. The cone has these little scales on it and these little weird leaves. But just for you to know, each cone scale has ovules on it. So, as I said, the microsporangia is going to be where meiosis occurs to make the microspores. Ovule, which is right there, is going to be where meiosis occurs to make megaspores. Eventually, those microspores will go through mitosis and things to make your pollen grain. Look, here's Mickey Mouse. And eventually, the megaspore will grow into a female gametophyte. So just so you know that you start with 4 megaspores, but only one of those megaspores will actually remain functional. And that megaspore, which is inside of the megasporangium, which is inside of the ovule, will grow that female gametophyte with eggs. So you can see here now it's female gametophyte because it has archegonia and eggs, and that's still inside of that ovule. Eventually, the pollen grain is here, Mickey Mouse. Eventually, the pollen grain will land on the tip of the ovule. That's what this is showing you here, and this is a close-up of that here. When the pollen grain lands on the ovule, this tube cell, pollen tube cell, will grow. And it will grow into the ovule and allow the sperm to reach the eggs, which is what is showing you down here. Pollen grain would have landed right here, right? Mickey Mouse would have landed right there. Pollen tube cell would grow into the female gametophyte plant, and the egg and the sperm would be able to be united. And when that happens, we've got our 2N zygote. And the ovule is now a seed. The only thing you should, so we left off up here, right, with our 2N zygote. Eventually that will grow. Only one of these will become a 2N zygote. Oh, right. Sorry. Yeah. Don't worry about it. So 2N zygote, and then ultimately after 2N zygote, you have this seed in the embryo. Sorry, you have a seed with an embryo inside, right? That's all you need to know from this. Don't worry too much about all these other words. Crazy. Get out of here. Embryo now is inside of that seed. Embryo is going to germinate out of the seed and grow into the new sporophyte plant, which we see here on the left. That young sporophyte plant will grow via mitosis to become the mature sporophyte, adult sporophyte, shown on the left here. Okay. We did it. It was so long. I had a tornado. I'm just like crazy lecture, but we did it. There is a quiz for this lecture on Canvas now. It's due by Wednesday at 6:30. Sorry, it's due by Wednesday by 6. And I appreciate you guys for hanging in there. I know this is a very long lecture. Trust me, the last lecture, which is next class, won't be as long. This is probably the longest one, but at least it's pre-recorded so you can take breaks. So with that, I'd like to just thank you guys for bearing with me and learning about plants, and I will see you next class for our last lecture of the summer. So have a good one. Take care.